Good afternoon. Good morning for you who are listening in from other continents, or good night, I have no idea. My name is Jaap Verwij. I'm the Managing Director of the Cancer Drug Development Forum, and it's my pleasure to welcome all of you over here in the room, and those of you who are participating online, to this workshop on the critical role of biomarkers in delivering drug development related precision oncology. I will only give you a few introductory remarks and then hand over to the chairs of the actual session. For those of you who are not aware, the Cancer Drug Development Forum is a non-for-profit organization actually operating from Belgium, and that organizes all of its meetings in the Netherlands, which provides a neutral platform for the stimulation and interaction between stakeholders involved in cancer drug development. So that basically means we have people that we invite and asked to come over from pharmaceutical industry, obviously, from the regulatory affairs, but also from the health technology assessment now, and last but certainly not least, the patient advocacy and the academics that are involved in the whole process, in order to have a debate on issues that we all share that need to be improved, or we would like to see improved, in the ultimate end to have a more accelerated development of the goods that really should get to the market as quickly as possible to give benefit to the patients. I have been on the program committee of this actual meeting, jointly with my colleague Mark Lawler from the CDDF as well. We both are members of the CDDF board. And we have had the honor to do this in conjunction together with Richard Fard from Eli Lilly, Ademi Santiago Walker from Johnson Pharmaceuticals, and Mariana Fitoloni from the Digestive Cancers Europe, Italy. Four of us are here. Mariana is online, and she will talk to you later online. Just a brief overview of the program for today and tomorrow. We will start with a session where we will be setting the scene of biomarkers, giving you some definitions, discussing definitions if we need to discuss them. Then we will move on to the regulatory part, the biomarker-based authorization, and the HTA part, the reimbursement. We will have a final session today discussing what if we would not be using the biomarkers, and there are various issues there as well. And then after we will have a welcome drink, there's a dinner on site here in the hotel at 7:30 that you're welcome to join. Tomorrow we will start a little later. This is to accommodate those that are calling in from the United States. For them it's 5:30 or so in the morning, 4:30. Still they get up to show devotion to CDDF meetings. And that will be a pièce de résistance, maybe, the in vitro diagnostics regulation and the issues around this. Then we will have a very brief wrap-up, and you can get some lunch to go home, take away lunch, or you can stay here and have your lunch here. Learning objectives of this meeting is how to define a biomarker, related populations within the health systems, how to align the conditions for involved biomarker assessment in the EU, the regulatory requirements for marketing authorization, and discussing the regulatory requirements for reimbursement approval after marketing authorization, so basically enabling patient access to novel drugs. Just to make you familiar on who's here and who's online, we have 44 people that are joining us here in the room, and we have 201 participants online, and you can see the mix of their backgrounds on the left part. 37 come from industry, 34% are actually regulators, 17% academics, and 12% patient advocates. And the HTA we have included in the regulators. For those of you who are online, this is the virtual platform that you can use, the Brella platform. If you're already listening in, you found your way already. On top, you've had the possibility to join the event. If you go just on the left a little bit below, you can schedule a networking event if you want to do so. There are people that have registered for this, and then you can find maybe somebody who you would like to talk to and make an appointment for basically an online meeting, which will be obviously not in the room. For those of you here, if you would like to participate, we always would like to spread the message of CDDF and our meetings. And Hyun Min, or Min, abbreviated as Min, is around from our staff and would like to talk to those of you who are willing to basically give a 2-minute recording of your experience here, what you felt was important, what you felt really stimulated the discussion and your participation to this meeting. So if you would be willing to do so, talk to Min. She's in the back of the room now. Which brings me to the actual start of the meeting, setting the scene of the biomarkers. And all I need to do now is finally introduce the 2 chairs that will actually be online and not present here. First one is Mariana Vitelloni from the Digestive Cancer in Europe. She is a laureate from the Medical and Pharmaceutical Biotechnology and obtained a PhD in genetic and human pathology and has been involved in many collaborations and cooperations within the American and Spanish institutions. And the other one is Jessica Vermeulen, who is actually in the Netherlands but has been hit by COVID, so she couldn't make it here, but still will be joining us online. The VP Clinical R&D of Janssen Biologics in the Netherlands and responsible for the clinical development of Janssen's portfolio targeting B-cell lymphomas, both in late stage and early development assets. I've known Jessica already for a long time, and she's very devoted, I can tell you, to the cause of CDDF as well. She attends all of our meetings. Unfortunately, couldn't make it today, exactly on one of the meetings where she's chairing a session. So without further ado, I'll stop here and hand it over to the the session chairs, Jessica and Juanita.
Welcome. Again, I apologize. I've been successfully dodging the bullet of COVID for the period of the pandemic. Unfortunately, now, just at the time when I was to chair this meeting together with Mariana, it hit me over the past weekend, and I felt it wasn't appropriate to expose you all to my sickness. So I'm sorry not to be in person, but I think we'll have a wonderful meeting online. I think that's what we've learned from the pandemic. So I'm very happy to chair with Mariana today this chair. So my name is Jessica Vermeulen. I work within Janssen. I joined Janssen in 2005 after a career in academics and as a postdoc to pursue my passion to develop new treatments for patients in need of treatment, specifically with cancer. I'm working on the Janssen's portfolio of targeting the B-cell lymphoma, as well as patients who are suffering from plasma cell diseases, specifically patients with AL amyloidosis. And looking at biomarkers, I think it's very important. So, Mariana, maybe you can briefly introduce yourself so people can hear your voice.
Thank you, Jessica. Happy to be here with you, even though I'm virtually there. I work as a patient advocate and medical and scientific project manager at Digestive Cancer Europe. That is the umbrella organization for patients affected by digestive cancers. My background, as Jap already said, is in science, and I see my function within the organization in creating this bridge between science and what is patient advocacy and supporting patients.
Thank you, Mariana. So today's we're setting the scene of the symposium, and the key objectives for our session is to understand how biomarkers are defined and their role in research and clinical practice, explore the practical relevance of the biomarkers for patients and healthcare systems, considering what their impact is on diagnostics and potential for personalized treatment, and also to recognize potential challenges and opportunities as well associated with the biomarker utilization in both the academic and industry setting. We have a great set of speakers, so let's dive into the discussion immediately. It's my pleasure to introduce the first speaker, Professor Nicola Normanno from Italy. After he graduated cum laude from as a medical doctor in surgery, he worked as a postdoc in the NCI in Bethesda and then specialized in oncology and hematology. Dr. Normanno is well recognized for his scientific career in translational science. He was awarded the Annals of Oncology Prize for Translational Research, and then was appointed as Director of Cell Biology and Biotherapy in the International Fondazione Pascale in Naples, where from 2006 onwards, he's also the chairman of the research department. Dr. Nicola leads the research group involved in both preclinical and clinical studies, in particular the identification of biomarkers associated with resistance and sensitivity. And Dr. Normanno's group has been the first to use the next generation sequencing techniques in clinical trials. And also he is, and we'll speak in depth about this, the liquid biopsy is very high on his agenda, and he's very much involved in the discussions about clinical diagnostics and the organization of national and international quality assessment for these tests. Nicola, thank you so much for being there in person, and the floor is yours.
Thank you for this nice introduction. If I can have the first slide. Well, these are my disclosure. Well, I've been asked to discuss the complexity of biomarker development, and I hope that this will not be too complex, but let's start from the scratch, so from the definition of what is a biomarker. So basically, cancer biomarker is a characteristic of the tumor that we can measure, that we can evaluate, and we can measure these characteristics in the tumor tissue, or in the blood, and this can be either protein, a sequence of DNA, or other characteristics. Now, what's important to highlight is these cancer biomarkers will provide us information on different issues. On the diagnosis of tumor, they will help us to define which is the prognosis of a tumor, so how much the tumor is aggressive, and then there will be also some biomarkers can help us to predict the toxicity of therapeutics, and finally, they will inform us whether or not the patient might respond to specific drugs. So based on this, it's quite clear that biomarkers are essential for precision oncology. We have many patients have the same tumor type, the same signs, the same symptoms, but this patient will have a completely different outcome. So using biomarkers, we can really better stratify patients to understand which are the patients who have more aggressive or less aggressive tumors, to understand which patients will respond or not to specific therapies, because basically biomarkers will provide us information on the biology of the tumor. This is why they are essential. Now, what I try to do in my talk is to provide you some examples on how we believe that the implementation of cancer biomarkers can completely change the approach to diagnosis and treatment of cancer. And let's start with the first point, early diagnosis. Now, there's little doubt that only early diagnosis of cancer may have a significant impact on survival. And this is true for any solid tumor. If we make a diagnosis of tumor at early stage, the survival rates will improve significantly. Unfortunately, we have available screening program only for 4 tumor types: breast, cervical, colon cancer, and some countries, and wholly in, let's say, high-risk individuals also for lung cancer. So we really need novel tools in order to identify early cancer. And among the others, liquid biopsy is really emerging maybe as the best approach for early diagnosis of cancer. Now, this liquid biopsy is a very generic term that we use to refer to the possibility to identify biomarkers in bodily fluids of the patients, and in particular in the blood. Now, in blood, we can find circulating tumor cells, we can find proteins that are released by the tumor, we can find nucleic acids, in particular DNA, that is released by the tumor, and this is a circulating tumor DNA, which is really emerging as a very good possible biomarker for early cancer detection. But this is quite challenging because actually we know that there is a correlation between tumor burden and the levels of circulating tumor DNA. So in the early phase of the disease, what will happen is that the levels of circulating tumor DNA are expected to be very low. So this is very challenging, make diagnosis at this stage. Now, there are a number of biomarkers that are now being explored. The most promising seem to be methylation signatures, because they are specific, they are sensitive, but they also get the advantage. They will provide information on the tissue of origin of the circulating tumor DNA. If you are developing a tool that is able to screen for different tumor types, it's not sufficient to know that the patient has a tumor. You need to know also where the tumor comes from. Now, there is also data suggesting that if you combine these methylation signatures with mutations, the copy number alteration, and maybe even with protein biomarkers, you can increase the sensitivity of the test. Sensitivity is important, but I want to highlight also the importance of specificity. This is a slide that I captured from a talk was given at the last ESMO Congress, and about the results of one of these trials in which when a tool was explored for assessing early diagnosis of cancer. And there was only 1% of false positive. But this 1% of false positive means there were 98 individuals that underwent several medical examinations, and I guess that a lot of anxiety deriving from possible diagnosis of cancer that actually at the end was a false positive. So we need tools that are very sensitive but also very specific. But the good news is that many companies are investing a lot of budget, a lot of money in these new tools, these new assays. So I hope that we will have some assays available in the next future. Let's move to prognosis. Now, the example that I'm bringing here is about early cancer. Many patients get surgery and they really are cured by surgery alone. However, some patients will experience a recurrence of the disease due to the fact that some tumor cells are still present in the body after we remove the primary tumor. And we refer to this as minimal residual disease because usually we are not able to detect this minimal residual disease. Now, what happens is that based on this, there are patients that are overtreated because if patient has no minimal residual disease and is receiving adjuvant therapy is an overtreatment, and there are patients that will be undertreated because maybe they have a minimal residual disease and do not receive adjuvant therapy or the adequate adjuvant therapy. So identification of minimal residual disease in early stage cancer is really important in order to provide a better personalized treatment to an individual patient. Now, there are a number of studies. I'm showing you the ones in colorectal cancer, but this is true for all tumor types that have clearly demonstrated that the identification of minimal residual disease by circulating tumor DNA testing after surgery is associated with much higher risk of recurrence. I mean, here this indicates the risk of recurrence, and you see the ctDNA positive patients have always much higher risk as compared with ctDNA negative, and this has been found in every tumor type. Are we ready? So this is a very strong prognostic factor, but are we ready to use this in our clinical practice? Now I try to summarize the open issues in this slide. On one hand, we know that almost all positive patients, MRD positive patients, will have a recurrence of disease. We have no proof that adjuvant therapy or which type of adjuvant therapy is really able to eradicate this minimal residual disease. So we need clinical trial to demonstrate that we can improve the survival of these patients. On the other hand, it's true that patients who are MRD negative do much better, but still some of these patients will have a recurrence of disease. And we don't know why. We don't know whether this is due to the fact that the test that we use, the assay that we're using is not enough sensitive, or it's due to the biology of the tumor. Because as a matter of fact, the majority of patients that will have a recurrence of disease will have a recurrence that is either local regional, in the brain, on the lymph nodes, and these are all sites that are shedding low levels of circulating tumor DNA. So I don't think it's a good idea today just not give a patient an adjuvant therapy based on ctDNA only, but we are working on clinical trials, so we'll have news also in this setting. But there are other types of biomarkers that can be used in this patient. For example, it has been demonstrated that the clearance of the ctDNA, so if you measure ctDNA before and after adjuvant therapy, those patients that are negative after adjuvant therapy do much better as compared to patients that are positive, so they have a better outcome. And this information will be really important because at this point, the patients that are still ctDNA positive could undergo maybe additional therapy. But it's been also hypothesized where we could use this clearance as a new biomarker, to use in clinical trials of adjuvant therapy to have an immediate sign of whether or not a new treatment is active. And finally, there are very preliminary data suggesting that with liquid biopsy in patients, these are actually data from lung cancer, we can identify the clones that will be responsible for the recurrence of disease even before surgery. Now, if this will be demonstrated, this means that we can identify the molecular alteration of the tumor cells that will be responsible for recurrence of disease, and we can provide to any individual patients a tailored and personalized adjuvant therapy. This would be a great innovation, of course, there's time to get to there. And finally, we learned how important are predictive biomarkers. Now we have available a huge number of new drugs, target therapies. These target therapies are able to selectively kill the tumor cells that carry specific genomic alterations that are the biomarkers that we need to test. And on top of this slide, you have the drugs that have been approved by the FDA in the past few years. On the bottom, you have the drugs approved by the European Medicines Agency. I would say that EMA is slightly lower as compared with FDA. Still, most drugs approved by FDA are also approved by EMA. And based on this, and this is a work we did with Mark Lawler, today almost 30% of patients with advanced cancer may receive a therapy that's based on biomarker testing. So this is not anymore an approach that is limited to few patients. Now that a huge fraction of patients can really benefit of biomarker-based therapy. Now, we know that if we want to identify this patient, we need to use NGS because we need, NGS is this technology that allows to identify different types of genetic alteration in multiple genes in a single test. However, still there's a discussion on which type of NGS we should use. At least in my country, I would say also based on the ESMO recommendation, it is recommended that in clinical practice you should use really only small panels that are targeting only the biomarkers that have been already approved for clinical practice. On the other hand, now we have available very large panel that we define as comprehensive genomic profiling panel. These panels sequence 300, 400, 500 genes, so they will provide you a lot of information on many genes and also information on complex biomarkers. But of course, these are highly costly and complex also to interpret. That's why the recommendation is to use comprehensive genome profiling in patients who failed the previous line of therapy, the standard therapy. So we wait till the patient becomes resistant before we offer comprehensive genome profiling. But this is really a good strategy. And to comment this, I'll show you some data from a clinical trial that we opened in Italy. This is a prospective trial of genomic profile, the Italian Registry of Action Mutation, the rational studies, and we really enroll patients in two pathways. In pathway A, we basically simply register the NGS data of patients who receive the NGS test in clinical practice or in clinical trials. And actually in this pathway, most of the patients were tested with the NGS panels. There were less than 52 genes, so let's say small panels. In the pathway B, however, we enroll patients who had no possibility to access to NGS, and we offer to these patients the possibility to have a comprehensive genomic profiling test. And what I want to just show you is that the patients in pathway B are more heavily treated, they have more lines, previous lines of therapy. So this is basically what you expect in the Italian practice. I mean, you offer CGP, comprehensive genomic profiling, when a patient has failed standard therapy. But what are the results? Well, we found actionable mutation in a large fraction of patients, but as in agreement with previous studies, only 11.7% of patients received a therapy that is matched to the genomic alteration. Now, the main limit that we and other trials faced is the problem of the availability of clinical trials with target therapies in which to put the patients. So this is for sure is the main limitation. But however, look what happened in pathway B. Almost 14% of patients died before the report was available. And almost 28% of patients died within 5 months, suggesting poor clinical condition. These patients, no way they could be enrolled in a clinical trial and most likely couldn't receive any kind of treatment. So the message that really comes from this study is that we want really to give an opportunity to these patients. We must not delay the introduction of comprehensive genomic profiling when patients have failed already 4 or 5 lines of therapy. I'm not saying that we should test with CGP all patients, but we not even wait. That they become resistant to any type of therapy, because at that point, the patient will not get any benefit from this testing. Another important information that is coming from comprehensive genome profiling is summarized in this slide. Now, when you use these large panels, basically you will sequence also genes that are involved in hereditary cancer syndromes, and here is the list of the 28 genes. Now, the new guideline for ESMO, the new guidelines extend this list to 40 genes, but at that time we had this 28 gene list, all genes that are somehow associated with hereditary cancer syndromes. And what ESMO recommends, when you find a mutation in one of these genes at a level of 30% or 20%, depending on the type of mutation, then you should send the patient to germline, to genetic counseling, because there's a high probability that that mutation could be germline. And when we apply this to our patients, we found that almost 12% of patients receiving comprehensive genome profiling had a potentially germline mutation. Now, when I show this data to my colleagues, medical oncologists, they tell me, okay, but why this is important? This will not make any difference for the patient. And this is true, but this can make a huge difference for the families. Because if by comprehensive genome profiling, we can identify families that have hereditary cancer syndrome, those are the family in which we can make a difference, in which we can have early diagnosis, individuals in which we can maybe do preventive surgery or preventive medicine. So I think that this is a kind of side effect of comprehensive genome profiling, but it may be very important and may have a very important impact on, the survival rates for cancer. And the very last part is about also the liquid biopsy, the use of the liquid biopsy in patients with advanced cancer. Now, the European Society of Medical Oncology released this recommendation, basically restricted use of liquid biopsy only for the identification of predictive biomarkers when tissue is not available, when tissue failed, or when you need a very urgent response. And actually, there are also indications to use as first screening when patients become resistant to therapy. And these are the only application that now we should use in our clinical practice. But as a matter of fact, we know that there are many more information that we can derive from liquid biopsy also in patients with advanced cancer. For example, we know that the dynamics of ctDNA is a very strong predictor of response to therapy, to chemotherapy, to target therapy as shown here, as well as also to immune therapy. So again, this could be a very early endpoint, in the clinical trials, but I have to disclose that also in our clinical practice, sometimes we use liquid biopsy for specific patients. Let me show one of our cases. This, for example, was a patient, a lady, who had a rare EGFR mutation. At that time, we did not have available any specific drug for this exon 20 insertion, so we usually put the patient on monitoring with the liquid biopsy. And when we see there is a sign of relapse, at that point, molecular relapse, we make a more intense clinical monitoring. And when we got the spike on ctDNA, or then at that point, we switch treatment, and when we're using a different therapy, we could have a molecular response that was correlated also to a clinical response. So this is really the power at the end of molecular biology when applied to our patients. Now, before I get to my conclusion, I will just highlight that while many academic centers have already available most of the technologies that I've shown to you, the problem is that the majority of patients in Europe have little access to biomarkers, have little access in particular to NGS tests, and also we have highlighted some issues on the quality of tests, because it's very important to get a biomarker test, but we must also ensure that the quality of tests is high. We don't want to have false positive, we don't want to have false negative, because they are really dangerous for our patients. So the conclusions. The message is clear. Biomarkers play a very relevant role for the diagnosis of tumors, to define the prognosis of our patients, and to predict the response to precision therapies. And we believe that all patients must have access to biomarker testing that are indicated for their disease. But we also believe, from an academic point of view, that the implementation of comprehensive genomic profiling and liquid biopsy in the diagnostic workup will increase the chance of patients to access to clinical trial, will help to identify families with hereditary cancer syndromes, and more generally will provide relevant information to improve the personalization and precision of therapies. My very last slide is to thank all my collaborators, and in particular IQM part for the work that we are doing on the access and quality of biomarker testing. Thank you very much for your attention.
Thank you so much, Nicola, for your great presentation. Please send in your questions through Slido. We'll keep a close eye on these, and we'll reserve all the questions for the panel discussion. It's now my pleasure to introduce the second speaker, Professor Mark Lawler, Professor of Digital Health, Chair of the Translational Cancer Genomics at the Queen's University of Belfast, and a board member of the CDDF. Mark is a very well-recognized international scientist with many papers on his name, key publications in high-impact journals, and also having received numerous national international awards. He's very active in the big data committees on several fora. He is leading the health data research in Wales, Northwestern Ireland, where the public health is informed through the use of big data. He's scientific director of DataCan. He's also the chair of the International Cancer Benchmarking Partnership that really uses data intelligence to support and underpin the improved outcomes for patients with cancer globally. And he has a strong commitment to patient-centered research addressing cancer inequalities. He was an architect of the European Cancer's Bill of Rights, and also addresses the inequalities and access in relation to cancer care, which formed the centerpiece of the development of the European Code for Cancer Practice. Last but not least, and especially in my condition, I'm very recognizing that, has been heavily involved in COVID-19 and the impact that it has on the research for cancer and the advancement of new treatment. Very active in the Build Back Better from COVID, ensuring that we don't give up on cancer research while COVID is impacting us, and also recognizing the impressive work on all the doctors that continue to deliver the patient-centric care and still continue the research. So it's my great pleasure, and thanks so much, Mark, for also helping us to guide the discussions later on in the room. Please welcome to the floor, and I look forward to hearing your presentation.
Thank you so much, Jessica, for your kind introduction, and thank you to everybody who's attending both here in the room and online for what I think is going to be a really great session. And Nicola has really kicked it off in a superb fashion and really has set the challenge to us to say, you know, how can we actually deliver precision oncology without the use of biomarkers, which is something I'm going to take onwards now as we talk about this. So I think we're primed for prime time here in relation to the delivery of biomarkers to actually affect clinical care, not in the research setting, but in the actual clinical setting and in the cancer drug development setting. And hopefully you'll agree with me at the end of this presentation. One of the things I think we have to really recognise, and also make sure that regulators, payers, etc., recognise, is the primacy of precision genomic testing in precision oncology. Because this is really critical. If we don't believe this, then I can leave now, okay? So really we have to look at ways in which we can build the evidence that shows that this is a case that we have to. And in fact, if we don't do it, we're actually failing our patients. And we really need to look at how we embed precision genomic testing into the real world of oncology delivery for our patients. And as Nicola said, we really have to implement this right across Europe. And we need to narrow, not widen, the inequality gaps that unfortunately we see for patients with cancer, particularly in Central and Eastern Europe. And the other thing that's really important is that cancer patient has to be at the centre of this process. They have to be embedded within the precision oncology agenda, and I'll touch on that slightly later on. So really in relation to what I'm going to talk about, I'm going to say, you know, let's really deliver something that's going to deliver for our patients. Because if we don't do that, I think we're failing our patients. We need to maximize the therapeutic response of the different medicines that we deliver and associated surgery and radiotherapy, but also minimize treatment-related toxicity. If we think back in relation to the sort of beginning of precision oncology, really these two, I think, were the poster children for the precision oncology revolution. Both Herceptin in ERBB2 positive breast cancer and imatinib mesylate or Gleevec for chronic myeloid leukemia. However, for both of them, really their prior use of a test to actually identify which patients are ERBB2 positive and which patients had chronic myeloid leukemia was key to the deployment of both the clinical delivery, but also in relation to the development of the tyrosine kinase inhibitors that were used in these treatments. So this drug biomarker combination really has become an essential component in delivering modern precision oncology. As Nicola said, this paper that we published together, we saw there were 70 approved drugs which required a recommended predictive biomarkers test at the end of 2019. That's now at over 100, and really it's not an insignificant number at all in relation to delivering part of our care. So it's critically important. This is not a sideline study. This is mainstream in terms of how we deliver for our patients. And really one of the things we need to look at is how do we make it more pragmatic? And I really like this work by Andrew Biancan and colleagues, the Glasgow cancer assays. It's not the only one, there's a number of ones, but these pragmatic assays that are used to actually deliver in the clinic precision oncology, the diagnostic testing that's required for that. I encourage you to read that paper in relation to that if you're interested in this area. But it's really critical that we deliver something that health systems can actually take on board. It's no point in delivering something that's esoteric that actually isn't deliverable to our patients. And patients, as I said, I want to put at the centre of the personalised medicine approach. And this was something we did with the European Cancer Patient Coalition a number of years ago. And what we said was do people actually understand what personalised or precision oncology is? And I talk both about members of the public, but also sometimes clinicians. And so what we did was we decided we would actually develop a guide for patients. It was an initiative of ECPC and a resource that really addresses the needs of patients. And the link there is at the bottom of this slide. Because what we wanted was that cancer patients would be empowered to look for precision oncology approaches to treat their particular cancer, so that they can actually make the best and most informed precision decisions about their personal care. So we produced both a booklet and a accompanying leaflet. It was co-created by patients and health professionals, and it addressed the questions that cancer patients may have about personalised medicine, including immunotherapy. Because it can be challenging. And it's important that we look at what are the uncertainties that patients may have, and sometimes they may find them difficult to articulate or to ask questions of their doctors. So one of the things we did, as well as different sort of sections explaining different aspects of personalised medicine, we also created a series of 10 questions that the patients may choose to ask their doctor. And our feedback on this has been really good in terms of people saying, yeah, that's really what I want, because there's sometimes I'm not sure what the questions I want to ask. And it's really helpful to have something that sort of guides you. And so we hope that this has made it much more patient-informed so that they can make the decision that's right for them, their own tumour. Not a general statement, but something that's relevant to them particularly. I'm just going to talk a little bit about regulatory challenges. We're really going to come back to this in the next session and also in section 4, because we certainly see there are challenges in relation to regulatory issues, particularly in relation to the In Vitro Diagnostics Directive, which has now been superseded by the In Vitro Diagnostics Regulation. And it is a potentially contentious issue, particularly in relation to laboratory-developed tests, which should be complementing the commercially manufactured diagnostic tests. And we are really worried as a community that this is going to limit the number of cancer biomarkers that comply with the regulation and therefore can be used to select, through a companion diagnostic approach, the best treatment for the patients. And this has come upon us over the last number of years. We've been warning about it. We're now in a situation where IVDR is going to come into full effect in May 24, I should say. And so we really need to act quickly and decisively. And that's why we've dedicated a session 4 in this workshop, specifically to IVDR. And the other thing that's really important is in relation to cost and value. Cost effectiveness of precision genomic testing, unfortunately there hasn't been a lot of studies done on that. We decided to try and rectify this to a certain degree, and we've looked at it in colorectal and lung, and others have looked at it in other diseases. And what we found was that actually precision genomic testing, particularly in lung, was cost effective in 100% of the scenarios that we modelled. And so both of the studies both underpin the need for robust health economic evaluation, but also can provide the evidence to underpin value-based care. And Peter will come back to this in session 3, which I think is really important, and also another study that I'm going to mention that we've just come out with. The other thing is in relation to making sure that precision genomic testing is available and reimbursed, is that it's available and reimbursed for everyone, not just the rich countries of Western Europe. So we need to look at a framework that allows everybody to benefit from the advances that are made in relation to precision genomic testing. And this systematic review is very good in this regard, really capturing some of the socioeconomic inequalities that are unfortunately seen in relation to precision biomarker testing and also precision oncology therapy. And then the other important thing, and again, this is very much part of session 3, what about the cost of not testing? People talk about the cost of testing. What about the cost of not testing and not actually delivering for our patients? This is really important, that we actually make sure that we deliver something that actually is deliverable, but also if we don't deliver it, I think we're letting our patients down. This is a study that we just published recently, the references there, where we looked at essentially all the precision oncology medicines, our first wave of precision oncology medicines and non-precision oncology medicines that are on the market. So 42 precision oncology medicines, 29 non-precision. I'm not going to go into detail on this because Peter will also touch on it later, but just some of the take-home messages. First of all, it's the most comprehensive economic analysis of precision oncology medicines in the marketplace. It also then shows that over 1 billion, and that's not a misprint, less in R&D spend has been used to develop a precision oncology approach compared to a one-size-fits-all approach. So it's actually cheaper at the R&D stage to deliver a precision oncology medicine rather than a non-precision oncology medicine. And also it shows, because the evidence has been not been developed and not been done in detail, to actually look at, is a CDX guided approach, can it deliver healthy benefits, but at a potentially affordable cost? And we've shown in this study that you can actually do that. So it's lowering the expense of clinical trial attrition rates that we're seeing, and also hopefully sparing the patients the ineffective treatment side effects. So I'm saying this here now, that treating patients based on their genomic makeup must be the direction of travel that we pursue going forward. And this study highlights how we can do this in a cost-effective way. And as I said earlier on, if we don't apply a companion diagnostic guided approach, I think, and certainly we as authors thought, that we're missing a huge opportunity to deliver the best, most affordable care to our patients. Just to finish off with, I'm just going to show just some data from another study that's been convened through the European Cancer Organization, which has a group on genomic tumour testing that I'm privileged to co-chair with Matti Apro. And we've really tried to bring together the community. We've had a series of meetings, a workshop we had in 2021, and we also had another one recently. And we came out with this report called Put to the Test, Empowering Genomics to Improve Cancer Care in Patients' Lives. And the key approach here is to provide guidance and information to both stakeholders, but also particularly important policymakers, so we can really embed precision genomic testing as a standard of care right throughout Europe. And we've identified 4 key areas where we had an ask. One was improved collaboration between cancer centres throughout Europe, so that we actually pool resources and improve patient access to precision genomic testing. The second was enhancing consistency in national approaches for both reimbursement and for access. And the third was to improve the collection and the use of data, and to include the cost-benefit considerations that I showed you in the last example. And the fourth, to ensure connected infrastructure and to address workshop needs. I recently presented at the European Society of Pathology meeting, and one of the things I said at it, and it's been quoted widely on Twitter afterwards, is no pathology, no precision oncology. So if we don't have adequate pathology resources in terms of people or resources, we're not going to be able to deliver precision oncology. And the other area that we said, we really need to align activity to relevant and existing European policies. So just to conclude in relation to where I'm trying to sort of bring us on this journey, and obviously to be backed up by other speakers in this session and in future sessions. We really need to move to a situation where precision genomic testing is absolutely embedded within our health systems. We're not there at the moment, I unfortunately have to say, and that's where we need to go. And everybody needs to work together on this journey to get us there so we can really deliver for our patients. We need to look at ways in which we can upgrade infrastructure. We need to spend, obviously, but we need to balance that against the benchmarks to deliver affordable care. It's no use if we're producing something that actually isn't transferable into the clinical setting. The IVDR issue is an issue that must be addressed. We certainly feel that we must really articulate that economic argument that I presented much more clearly and robustly, and that actually economic assessment should be part of all the different studies that are being performed in this area, and also the importance of education. For patients, as I showed you in relation to the European Cancer Patient Coalition study that we did and the booklet that we produced, but also for healthcare professionals, for health policymakers and for healthcare providers. And obviously that's part of what we're doing here today and tomorrow in Amsterdam with this biomarker workshop. Critically, we really must avoid a multi-speed Europe that leaves many patients behind. This can't be precision oncology for the rich. This has to be precision oncology for all. A quality agenda must be there in place so that we can really narrow rather than widen the cancer disparities that unfortunately exist across Europe. So just in conclusion, I think we have a huge opportunity within our grasp, and I suppose what we really need to do is grasp that opportunity. Thank you very much.
Thank you, Mark, for this great presentation. Food for thought, definitely, and rightful attention for the disparities and also the IVDR, which tomorrow is indeed a great session. We're moving to the last speaker of our session, which is my great colleague. It's my pleasure to introduce her, Dr. Ademi Santiago-Walker. She's from the US. She traveled all the way. She's the head of prostate cancer and colorectal cancer translational research for the oncology therapeutic area at the Janssen Research and Development. And in the Oncology Translational Research Organization, Ademi leads the team of biomarker scientists who develop and drive personalized medicine and clinical research strategies to really enhance the development of oncology or therapeutic options for patients with prostate and colorectal cancers. Ademi has led the biomarker and real-world evidence strategies for the FGFR inhibitor erdafitinib, as well as Balversa, supporting FDA approval for patients with locally advanced or metastatic urothelial carcinoma with susceptible FGFR3 and 2 genetic alterations. And this approval actually marked Janssen's first approval of a targeted therapy with a companion diagnostic. So she's the right person to kind of speak about her experience and also the application specifically of real-world evidence to support that FDA submission. So Ademi, thank you so much for coming, and the floor is yours.
Well, thank you everyone for attending today and for the opportunity to speak from the organizers. Today, I'd like to walk you through the industry perspective on biomarker development. Specifically, we'll talk about the types of biomarkers and the application in drug development, the biomarker trends that we're seeing in FDA approvals, and then also walk you through the example of erdafitinib, of which was a biomarker-driven asset. So multiple types of biomarkers, disease-focused and response-focused available to us. For disease-focused, we have susceptibility biomarkers for healthy patients, their risk of actually developing a disease, diagnostic biomarkers to actually confirm a disease diagnosis, as well as prognostic biomarkers to identify how patients will perform in their disease journey. In terms of response biomarkers, we have predictive biomarkers to identify patients who might respond best to a particular therapy or potentially have an adverse event as well. Pharmacodynamic biomarkers are routinely used to determine that we are actually hitting the drug target as indicated based on mechanism of action, as well as longitudinal testing with monitoring biomarkers. As mentioned previously, interest in minimal residual disease, for example, and safety biomarkers to let us know if an adverse event is coming or has occurred. So this is used, all these biomarkers are used across the development spectrum from discovery all the way through to clinical implementation, particularly biomarkers to define what targets we should be going after, help us select a particular drug candidate amongst many based on modulation of a particular biomarker, confirming mechanism of action, target engagement, et cetera. And importantly, important for today's discussion is for patient stratification and patient identification. So this is an excerpt from a recent paper showing trends in oncology drug approvals for the last 22 years, from January 2000 to October of 2022. So of 573 drug approvals of 206 distinct drug products, This group will be happy to see that the bulk of approved drugs in that time were targeted therapies, so drugs that were designed to act on a particular biological mechanism. Only 9% of the drugs in recent time are agnostic of a particular medical or particular biology. So the highlighting here that the focus here in the past approvals in the past years were on drugs that had a biological rationale approach. Within those, approximately 39% of drugs were those with targeted therapies. For the most part, that you see that in panel A and panel B, most of the patient selection is positive selection, meaning that the patient actually has the biomarker, though there are some cases where patients were excluded based on biomarker status. In panel C, you'll see for these targeted drugs, approximately half of targeted small molecules and 1/3 of targeted large molecules have run their trials with patient selection. Importantly, what this means to us is what effect has been had by having these targeted therapy approaches or personalized medicine approaches. Well, overall, this is a publication that looked at the probability of success from entry into the clinic through to approval of drugs, whether or not patient selection was applied or not. And if you look at the far right, across the phase from phase 1 through approval, there's a twofold increased likelihood of success. Success meaning approval of the drug with drugs that had patient selection strategies. This is largely driven by the stage moving from phase 1 to phase 2, where you see the most of this effect. So with the positives that come with that, there are both opportunities and challenges with biomarker-driven studies. So as mentioned previously, higher probability of success, and as Dr. Lawler indicated, less likelihood that we will be giving patients who would not respond exposure to potentially to therapies that might have some potential side effects. This is also thought to lead to faster drug development and with significantly less cost. However, there are challenges associated with biomarker-driven studies. Importantly, the validation of the biomarker. There is a large onus to develop the scientific basis, not the scientific rationale, but the data to prove that this biomarker is sensitive to the drug and driving disease. And so confirming its predictive and prognostic value, especially in association with a particular therapy. The development of a diagnostic test is no small feat. Most pharmaceutical companies are not test development companies, and so it requires a lot of partnering to be able to determine how to best develop a test in line with the clinical development, so that at least in the US, where a companion diagnostic is required for targeted therapies, that test must be available at the time of the drug approval. So it adds another layer of complexity to the drug development process. And as Dr. Lawler highlighted, really making sure that test implementation and adoption occurs. So there can be drugs available, but if the patients aren't getting tested and therefore do not have access, that is a disservice we're doing to the community. So I'll talk a little bit about the experience that I had working on our erdafitinib program. So erdafitinib is a targeted FGFR1 through 4 inhibitor that was developed in bladder cancer. So fibroblast growth factor receptors are important in cell growth and differentiation. And given their important role in those key aspects, it can be co-opted in the context of cancer by having constitutive activation via multiple mechanisms, whether it be gene amplification, mutation fusion, or even ligand overexpression. So I'd like to highlight erdafitinib is an example where biomarkers were used successfully in the drug development process in two ways. One is we used a pharmacodynamic biomarker to optimize and individualize patient dosing, and two, patients were selected upfront for treatment with our therapy. So in terms of the pharmacodynamic use, FGFR is important in phosphate regulation. So it's expressed in the renal proximal tubule of kidneys and is responsible for removing phosphate from the blood. So as you would expect, when you're inhibiting FGFRs, one of the natural consequences is an increase in phosphate in the blood. So we knew this to be a pharmacodynamic biomarker and studied it in our phase 1 trials, and also found that there was an association between reaching a certain level of phosphate, for in this case was 5.5 milligrams per deciliter in the blood, and a response to erdafitinib, so such that those who achieved at least a 5.5 milligram per deciliter soluble phosphate level were associated with better response. So therefore, we use that information to look at individual patients on treatment. Each patient started off with 8 milligrams of erdafitinib dosed daily, oral dosing, and a phosphate level was assessed 2 to 3 weeks after starting treatment. For patients who had not received that 5.5 milligram per deciliter threshold, we increased their dose to 9 milligrams daily. This is also how we continue to use the drug even as it's been approved. Importantly, as well, in addition to the pharmacodynamic biomarkers, we identified patients upfront for treatment with erdafitinib. What did this mean for us? So we had 3 questions in front of us. Which FGFR gene was going to be relevant in bladder cancer? What FGFR alteration types should we be targeting to select patients, as well as what specific alterations? So FGFR is a family of 4 receptors, so it's important to understand what was the expression level and relevance in bladder cancer. And in terms of what particular alteration type, we learned a lot from our phase 1 study. So in phase 1, we enrolled multiple mechanisms of FGFR alterations, whether it be amplification, mutation, fusion, et cetera. And in the context of that phase 1, saw early on that mutations and fusions were particularly sensitive to erdafitinib inhibition. We used that information from phase 1, along with a computational biology landscape assessment of what alterations looked like across the bladder cancer spectrum, to identify 4 specific mutations in the FGFR3 gene and 2 mutations in 2 fusions in the FGFR2 or 3 genes as eligible for the study. And this ultimately became the diagnostic panel that we used not only for patient selection, but for the final companion diagnostic. In terms of validation of the biomarkers, the results of the phase 2 study in bladder cancer were sufficient to have an accelerated approval of erdafitinib in April of 2019 with 40% response rate in bladder cancer. So this highlights an example of where we've used it both for individualized dosing as well as personalized medicine and identifying the patients based on biomarkers in their tumor. A recent, semi-recent change in the way these studies are done is the advent of histology-agnostic drug trials. So the concept here is that instead of doing disease-specific trials in prostate, bladder, et cetera, the concept here is that the biomarker is the driver and it will have relevance independent of the tumor type. So treating based on the biomarker rather than the tumor type. This has been especially important in being able to perform studies in rare diseases. So for many of these biomarkers, they're found at very small frequency, and the ability to perform a clinical trial in a rare disease may not be feasible, especially considering that some of these biomarkers are only present in approximately 1% of cancers. However, so we have seen the approvals in 2017 with pembrolizumab and MSI high or DNA mismatch repair tumors. And since then, several have been approved. Now, this is important, again, in rare diseases because looking at a study that was done looking at the genomic landscape of patients using the AACR Gini database identified that 16.5% of patients have an alteration that has shown some clinical benefit in another tumor type. Importantly, the underlying hypothesis for these studies is that indeed the biomarker is a driver of disease across tumor types, and that the response and resistance profiles will be similar across those tumor types. So with that, I'd like to walk you through briefly the experience we've had with erdafitinib in our first histology agnostic trial, which is the Ragnar trial. So importantly, FGFR is one of the most frequently altered genes in cancer. Example here, looking at the FGFR expression profile or FGFR mutation landscape for FGFR3 or the FGFR2 gene. Each one of these circles represents a distinct mutation all along the FGFR gene, and the ones that are most frequently observed are above here. So clearly a lot of different FGFR alterations. So the task that was put on us was which are the ones that are likely to be predictive to outcomes with erdafitinib. So multiple studies, which I won't go into here, were performed to identify our target list of FGFR alterations for the study. On the left is the design of the Ragnar trial. So patients with any solid tumor could enroll in the trial ages 12 years and up. We identified a panel of specific FGFR mutations that conferred eligibility, and any FGFR fusion with an intact kinase domain was eligible. The dosing strategy was similar to our bladder with the individualized dosing based on serum phosphate. So in this study, 217 patients were enrolled, and I'd like to take the time here to discuss some of the key challenges, which was the ability to enroll patients and what it takes really to execute on these biomarker trials. So FGFR frequency across tumor types can be as high as 20%, but less than 1% in some tumor types. So that confers a challenge, and which is seen here by the number of patients it took to enroll these, number of patients screened to enroll patients. We used a combination of central testing, which was for patients who did not have genomic testing available to them already, and local testing. So the local testing was based largely at academic institutions which perform next generation screening as part of routine care. And so therefore those patients were already known to be FGFR positive. In terms of the central screening, where their positivity was unknown, 11,000 patients were screened to be able to enroll what was essentially 42 patients on trial. So it is something that needs to be considered when developing these types of trials, and something that we're learning to adjust to. But certainly the availability of next generation sequencing, as mentioned earlier, is going to be critical in providing access to patients, these therapies. So as the results of this trial, not all tumor types showed a response. However, we did see objective response of 30% across 16 distinct tumor types. On the right is an example of the tumor types with the highest response. In pink is the objective response, and in blue is the disease maintenance. And so the concept here is that, yes, we did meet our endpoint in this study, but it did take quite a bit of screening to be able to achieve that. So in total, what hopefully you've taken today is that biomarkers play an important role across the drug development spectrum. They have the potential to accelerate patient access to safe and effective therapies, and that targeted therapies are becoming the norm. Importantly, they must be validated for their use, which is a big part of what we are trying to achieve in our group. And patient selection approaches increase the likelihood of success, but may also increase complexity. And importantly, biomarkers have an integral and essential role in drug development. Thank you.
Thank you so much, Ademi. I think we're now ready for the panel discussion. So may I please ask the panel to take a seat. I hope there will be chairs at the podium. I can't see it, but that usually is the case. Yeah, there they are. I see them, but not filled yet. So Ademi and Mark and yeah, wait.
I'm going to take questions in the room just because of both of our co-chairs are online, but maybe just to start off with, maybe if our co-chairs want to ask a couple of questions to get the conversation going, and then just if anybody who wants to ask a question in the room could put their hands up, and then if they could go to the microphone, and just when you're doing that, if you could just give your name and also the organization that you represent, if that would be okay, that would be great. And then those who are online, if you can use the system to send in questions, they'll be asked then by our co-chairs. Okay.
Thanks, Mark. So maybe I can kick it off, and then usually there's plenty of people asking other questions. So maybe I can start off with a question to you, and Ademi and Nicola may also comment. But how can we make best use of the genomic tests in routine clinical practice? Because I think both you and Mark, said that it's being done. There is routine testing for most tumors. Yet if you look at the heroic effort that Ademi described to screen 11,000 patients to identify the right patients for a trial, that makes it quite difficult. So what is your take on how we can best make use of the genomic testing that we're trying to get in routine clinical practice to advance the development of new treatments and making them available?
Do you want me to start? Okay, so maybe I can start. I mean, I really liked Andrew Biancon's use of pragmatic cancer assays. So the Glasgow assay is one example. Now, it's not the only one. There are other systems and other health systems taking up that type of approach. But it sort of very much then leans towards saying, let's use the evidence that we've generated to develop something that actually can be put into routine practice in the National Health Service in the UK, for example. So I like that type of approach because it's very much thinking about the end game. And we touched on this in your presentation where you said, we do need something that's actually deliverable to the health service. And that's not to exclude the approach of looking at sort of larger panels, etc., which obviously have their place as well. But I think if we can get embedded pragmatic assays into health systems, that will be a big success, and it'll be a big success for our patients.
One thing in terms of the size of the panels, one thing from the perspective of our team when we were thinking about what test to implement, given that FGFR doesn't have a high frequency across a number of cancers, we did aim to utilize a panel that did have a wider selection of markers, because we're thinking about it from the perspective of the patient is providing the precious tumor to be able to do this testing. There might be at max a 15% chance of them having the alteration, but at least they can get more information that might direct their next therapy with those wider tests.
Well, that's something that in many academic centers we are switching to use a comprehensive genomic profiling test, which is okay. But there are several issues that need to be solved. One of these is that sometimes the enrollment of patients requires a central test. And this is an issue because sometimes you have only very little amount of tissue available. So we have to find a way in which the academic centers are validated, but then, the companies must accept, the test has been performed locally. Otherwise, it will be very difficult that, we have to go through new biopsy for our patients. And sometimes you have to decide whether to run the test in-house or send out if the patient is eligible for a clinical trial. But sometimes, the eligibility of the clinical trial does not provide me then any information that I can use if the patient is not eligible for enrolling in other trials or to use just in a routine clinical practice. So this is something that needs to be solved. And because this goes through centralization and some kind of validation certification of the centers, because this is something that we have to work together. This is the interest of the academia, is the interest of the patients, as the interest also of the companies. Because if we find a network of labs that are certified and high-level quality academic labs, at that point you can accept the test. I understand that the situation with FDA is different than EMA, but we have to find a compromise. Otherwise, it would be very difficult also to enroll patients in Europe for this. The other issue is that we are using academic centers, these large panels that we have now a new law in Italy that is supporting the use of panels for molecular tumor board eligibility. But at the end, it's not clear who is going to pay for that, because are we using funds from the health system in order to support the enrollment of patients in a clinical trial of a company, and this is an issue that needs to be, maybe there has to be some participation in the cost from the companies if you want really to screen patients, and that will make it easier, the enrollment into the clinical trials. And then the last point that I would like really to hear also the opinion of Mark, and then we have also maybe in the next sessions, really, we do believe that comprehensive genome profiling can make the difference, but the current HTA does not take into account all the advantages that come from comprehensive genome profiling. If you look only at the cost of the test, of course, the cost of CGP is higher than the standard test. But if you do CGP, you can have a lot of information, you can put patients on clinical trials, you can maybe identify patients with germline mutation. So we should find also a different way of HTA in order really to assess which is the value of an extended testing and why we should implement this into the health system.
Yeah, I think you're very correct, Nicola. I think one of the problems at the moment is we've got a new system, and we never, in relation to comprehensive genomic profiling, and we maybe haven't quite worked out how do we measure the value of that, like right across the piece. So I think one of the things we need to do is look at that and how best do we do that. I think the other thing that we need to look at is how do we, one of the challenges I face when we're trying to make decisions in relation to cost saving is cost saving in relation to diagnostics and cost saving within relation to treatment are in two completely different areas, even within the hospital. So I might be trying to say, well, this will save you money. And they say, well, it won't save me money. It'll save that department over their money, but that's not saving me any money. So I think we need to think more broadly rather than being siloed into little boxes as to who's paying for a particular part of it. I think that's part of that sort of linking the biomarker to the treatment. So that's sort of the companion diagnostic approach needs to be looked at, not just in terms of the delivery and the science associated with it, but also the economic assessment of it, the way in which we deliver into healthcare. We need to think of both of them, and the problem is they go down different pathways at the moment. And even if we think about it, we have a diagnostic pathway for cancer patients, we have a treatment pathway for cancer patients, but we need to look at ways in which they can sort of interact with each other more. Question from the audience there. If you just approach the microphone there.
Yeah, hi.
We also have a question in slide after.
Okay.
Perfect.
Yeah, can you hear me okay? Yeah, so hi, I'm Richard Vart. I'm from Eli Lilly. So yeah, right, Mark, really great call for action. I thought a really great setup for the program. And the latest one on efatinib was really interesting. And it was interesting in the fact that then when you did the study for the tumor agnostic, because, one vision is that one day you come in a patient, you screen it on a genomic thing, you identify your driver mutation, and off you go. You know exactly what to treat. This is the vision that I guess we are all going for. Why do you think it was that basically you had this FGFR mutation, you had these tumors, and yet some didn't work and some did? Is it that it wasn't the right driver mutation, or was it that maybe a mutation that you selected up front in your panel wasn't right? What do you think it was, and how do you think we can ever get to that state?
That's a great question, and one that we're actively working on. I think it's a lot of the things that you discussed. There could be specific mechanisms of resistance in a particular tumor type, also to whether it is a driver, because these alterations are not found alone. They're often found in conjunction with other alterations. We found in our first trial that we didn't necessarily need to exclude based off of other genomic alterations, but that was a very bladder-focused trial. When you're talking about any solid tumor, you get the whole spectrum and plethora of co-alterations. And so that's where the evidence of the validation becomes critical, and the number of patients you need to be able to confirm that. And the idea is that you would get sufficient to be able to see that it is a validated biomarker, but will everyone respond? I don't think we could necessarily anticipate that.
Interesting, thank you.
So there is a question for Professor Normanno about the need of high-accurate tests. Do you think we should have strict harmonized acceptance criteria across biomarker, or decide case by case?
Can you repeat? Sorry, I didn't
Yeah, sure. About the need of eye-accurate tests, do you think we should have strict, harmonized, accepted criteria across biomarkers or decide case by case?
Well, I would say that the criteria are similar for all different types of biomarkers. And basically, I mean, you need to at least have first an accreditation, an ISO accreditation. Now we have some specific technical accreditation. We know that many labs, most labs in Europe are not accredited. And then is the participation to EQA schemes. We are working a lot on the organization of external quality assessment schemes. These are, independent bodies that prepare mock cases and send the material through the labs. The labs will test blindly, of course, in a blind fashion, and then send back the results to the provider. And by doing this, we assess the quality of the lab. I mean, this is the way we. And this is a very important process because we can discover errors that otherwise, you are not able to discover in your process. So basically, I'm involved in the International Quality Network for Pathology, and we are trying to provide a wide portfolio of EQA schemes, and in particular, when there's a new biomarker, we work in order to prepare the new EQA schemes so this is available. And this is very important also to start collaboration also with companies, because sometimes you get that the drug is approved, you need to have the test and there is no test available, and then when the test is available, it will take maybe another year or so before the EQA scheme is available, so before the labs can really verify the quality of their testing. So we need to anticipate this. When there is a drug that is really almost arriving to the market, maybe 1 year before at least, we need to start to work to put in place a quality system that includes the external quality assessment schemes, includes the recommendation for testing, so that we make sure that the quality of testing is high. But these are the two key points that we need to have. ISO accreditation and participation to external quality assessment schemes to ensure that the quality of tests is high. We have seen that when there's the introduction of a new biomarker, the error rate is high, and in particular when the new lab starts testing, the error rate is quite high. We don't want really to have this.
Thank you.
Another question in the room.
Hello, my name is Sarah van Waalwijk from the National Committee of Payers in the Netherlands. I have a question regarding continuous biomarkers, incremental biomarkers. So if you're doing a trial and you want to find the subgroups that react, response to a treatment, so the responders. So what do you do with markers like PD-L1 or human recombination, homologous recombination deficiency? So when you're dealing with this kind of continuous markers, I always seen nested biomarker, nested analysis, subgroup analysis, which is not really helping us as payers. So what would be the best way to do such studies and do the analysis?
Thank you very much for the question. Nicola, would you like to take that
Well,
starting point?
to identify cutoff in these cases is very difficult, even because there's a huge variability between tests, and this is another issue. Yeah. So when you put these drugs on the market, labs will be using different types of assay, and we are also working trying to find similar cutoff between the different assays. For example, we are working this on HRD. We are done for TMB. I think that sometimes the cutoff is good to have a pre-established cutoff in which that you explore into the clinical trials, but we have also to accept that sometimes some retrospective analysis can bring some new data. And of course, I understand that for payers, a retrospective analysis is not sufficient, and so we have to go again to a new trial. But we have to, with these continuous biomarkers, there is an issue that is not easy to address for the clinical trial, but also for the clinical implementation. I guess this is something that also the pharma companies have to address.
Yes, I mean, in deciding which biomarkers to pick, we certainly have to look at what's already been done. The cutoff establishment is really a challenge, and I think PD-L1 is probably the best example of that, with the number of retrospective assessments that have been done to try to harmonize that. But yes, it is something that it's a challenge.
We have another question from the online audience. What is the impact of biomarker quest on the development time of a new drug as opposed to all-comers approaches? Do we really always need biomarkers first? Who wants to take?
So there have been studies showing that biomarker-driven trials do result in a quicker signal and therefore, quicker development process. And so I'm sure, Dr. Lawler, you have a slew to talk about.
Yeah, no, definitely. I mean, I think one, we see differences in terms of timing. So certainly there are a number of studies now that have shown actually by using a biomarker approach, you not only get to the point of success quicker, but you also get to the point of failure quicker. So if you want to fail in a clinical trial, you want to fail early. So in terms of using biomarkers allows you to actually show the drugs that are not going to go forward. And that's equally important because we don't want to be bringing potential medicines all the way to phase 3 and then finding that they're not working. That's completely no use to the patient, no use to the payer, no use to the company that's developed the drug. So it's both in terms of identifying positivity or a signal earlier, but also identifying those which are not going to be candidates to deliver to patients routinely. And the second thing obviously relates to the cost and the study that I showed you and that Peter will refer to as well in session 3 really showed that using a biomarker-driven approach not only gives you an earlier answer, but also gives you a cheaper answer in relation to actually identifying which drugs potentially will go forward. And I think that's really important, being able to show both a cost benefit as well as a time benefit, because that's essentially when we're developing new medicines, we want to do it quicker, more effectively and cheaper. And we feel that certainly the evidence is now accumulating that that can be done.
Maybe related to that, Mark, Jaap Verwij from CDF. How much evidence is there actually for the opposite side? So you're discussing starting a drug based upon a biomarker data. How much evidence is there for early stopping a drug for reasons of a biomarker? ctDNA could, at least in theory, serve as that purpose, and stopping prior to whatever resist progression might be cheaper, at least from a HTA perspective, than just moving on with a drug that really doesn't hit the target anymore. So how much evidence is there?
And not as much evidence as for the latter in relation to identifying the positive, but certainly there is some evidence accumulating. And one of the things now is people are starting to look at approaches to be able to do that. So that sort of identify the non-runners at an earlier stage. And I think we should be focusing more on that as well, because it's another way in which we can use precision oncology and biomarkers to actually identify which approaches should go forward or not, as the case may be. I think it's really important to the industry that if you're going to fail, you fail early.
Yeah, thanks.
The issue is also whether the regulatory agency will accept as a new endpoint, a ctDNA endpoint. On the other hand, this would be also good for patients, because if you know that a new therapy is failing, at least you have the time to switch on the standard therapy. So I think there are great advantages, but this has to go also through some kind of agreement with regulatory agencies, I guess.
Well, I'm working in GIST. I have been working in GIST frequently, and there are 5 drugs now that are registered for use. And their ctDNA actually predicts that a drug is going to fail, and also predicts that another drug might help. So an early shift might be beneficial and save us at least some cost of a drug that is no longer effective, giving that to a patient.
Yeah, for the shift, I'm not sure, because we have some experience also in lung cancer, and we know that when we start to see resistant mechanisms, the patient still will have a response to the first drug, and sometimes even for several months. So, I mean, this is to be really demonstrated for the shift, but the dynamics is really, really important, because if you don't see any clearance of ctDNA, the patient is not going to respond. I mean, that's for sure. The point is it going to be scientifically feasible also to put in a clinical trial? What about regulatory agencies? Do you have any experience?
Yeah, so certainly from the biomarkers perspective in our team, we really champion the inclusion of ctDNA in our trials. It's a matter of building the sufficient level of evidence to be able to make a convincing argument. So from our team's perspective, we certainly want to have the data. When it comes to whether it be an adaptive design or that kind of trial, I just don't know that we're there yet to have the data to be able to implement that.
Thanks.
I would agree. I think there are some examples where perhaps the evidence is there, but I think we still need to accumulate more data for ctDNA to be used routinely to actually be a driver of go, no go, if you like. Axel?
Yeah. Hi, my name is Axel Glasmacher from the CDDF as well. And I agree to everything that have been said, but I would like to pinpoint a little more of the hurdles that there are. So one is obviously, and it has been mentioned, the high cost of screening. If you have a biomarker in 10% of the population, the number of patients you have to screen is extremely high. And then can you do it with local tests? Probably you cannot, because you need a validated central procedure. But can you use that if it needs to be a biopsy? Can you use that for other information to guide patients to other trials? So I think everyone would agree, doing cheaper drug development, failing earlier, it's a perfect world. But there are these hurdles that I think we have to address, and I would love to hear the panel comment a bit more on that. Thank you.
Sure. I'll start. I mean, certainly in terms of developing the test, especially if one hasn't already existed, is a huge hurdle. As mentioned, most of the companies are not in the test development business, but identifying the biomarker, but executing on that in the test has been a challenge, and with significant costs. There's multiple changes that happen. It's not as straightforward as laid out in the slides. There's years of twists and turns, and to be able to identify the right population, but importantly, the test to be able to do. In terms of the screening, absolutely. One of the challenges, one of the ways that we've thought to get over the challenge is to try to, as much, provide a high stringency of criteria of what we would accept for local testing, that it would be CLIA certified or regional equivalent. But those are all very real challenges.
Yeah, I fully agree. I mean, as I told before, we need to organize a different system in which sometimes even to send out the samples, and the patient has to wait 2 to 3 weeks before they get the result from central testing, and sometimes they can't really wait. So when it's possible, we do in parallel 2 tests. We test in-house, and then we send also out. But most of the times, patients then are treated based on the test that we do in our lab. But if we can get to a level of certification of the labs that is accepted by regulatory agencies, because I understand that's the limit, then we can really optimize the approach, because in this way, for most patients, and if you think lung cancer or other tumor types, we have very little amount of tissue to test. And most of the times, the tests that are centralized are not really adequate to test these very small samples. I mean, we have developed much better tests in our labs to address the issue. The only problem is certification. Maybe we need a different kind of interaction between regulatory agency, academia, and industry. We have to find some kind of agreement to move the field.
I think just Axel, the other important thing is we do need to invest. I mean, I've given some data that shows cost effectiveness, but that doesn't mean it doesn't cost. And so we do need to invest in comprehensive biomarker testing. Industry needs to invest in that as well as governments. So I think it's important that we recognise that there is an investment required, but that investment will pay back then in relation to better approaches to deliver for cancer patients. Another question in the room.
Hello, my name is Dirk Hemming from Daiichi Sankyo, and I got a question regarding EQA schemes. You called for the action to participate in EQR schemes, and you know that there are a lot of different EQA scheme providers and a lot of different EQA schemes, even for one biomarker out, and also they have different criteria and so on and so on. Do you see the need for a pan-EU EQA scheme agreement or something like that?
We are already working on harmonization of EQA schemes. The International Coordinating Network for Pathology includes the most, if not all, European EQA providers, but also providers from Canada, from the United States, Australia, China, Japan. And what we do, when there's a new biomarker, we are really working on the harmonization. We want to define the same criteria for the assessment of the scheme. We try to use similar samples. So we are really already doing this work because we noticed at the beginning beginning, 10 years ago, there were some labs that were participating to different EQA schemes. And then by chance, at least they could pass one. Now we are doing this work. But the true problem is that in the majority of European countries, it's not mandatory to participate in an EQA scheme. And if there is a poor performance, they will continue to test. See, this has really to be changed, because otherwise, we do a lot of work, but we don't see the results. On the other hand, I can tell you that from experience that we had in Italy and other countries, we have really seen that by introducing EQA schemes, you can raise the bar and the quality of the biomarker testing improves over the time. So it really works. There are some slight difference between the providers. This depends also on the fact some regional provider or national provider can use true tumor tissue, but if you are doing a broad program of participating 100 labs, then you need to use a synthetic material, but we are trying to harmonize this as well.
Okay, thank you.
We have now a question from the audience online for Dr. Santiago Walker. On what factors would you base a decision to pursue a histology agnostic medicine?
So I think that would be based on the presence of the alteration across whether it be solid or heme tumors. That's the primary criteria, is do you see this alteration routinely across multiple tumor types? And then if so, what context do you see it in terms of co-alterations that are also present? And has there been an indication that this biomarker is indeed one to pursue? So pursuing a histology agnostic trial, I don't think would be the first approach. There would need to be a prior trial that showed that the biomarker was indeed predictive of outcomes.
Thank you. Anything else, Nicola, from you?
Yeah, actually, we have already sufficient evidence that some co-alterations are limiting the effect of some of these target-based therapies. And actually, the point is that if you don't do comprehensive genome profiling, you don't have the picture. That's another advantage, because the heterogeneity of tumor will dictate whether the patient is responding or not. Now, if you have different, only one option, this will not make a difference. You will treat the patient anyway. But let's think what happens now, for example, for EGFR mutant lung cancer patients. You can use EGFR tyrosine kinase inhibitors, or combination with antiangiogenic, or combination with chemotherapy. How you stratify patients. Commutation most likely would be a key. So if you have the whole picture, you can better stratify patients and you can make a better decision. So that's why we need to move to that.
Another question in the room from Ruth.
And well, to follow up on that point, we had a CDDF ran a workshop on histology independent, as the EU phrase is now, and the white paper that the team of us produced from that we've just submitted to Cancer Treatment Reviews, so there's a position paper on that. But no, I was following up on the previous question really to think, are we at a point in Europe and to actually have some kind of harmonization of what we do in terms of precision medicine, in that we've all been doing testing. We now do cfDNA on all patients referred to the phase 1 unit. But we know the benefits with Mark's paper of both for industry on getting the registrations, but more importantly for patients treating with the right drugs and avoiding toxicity if it's the wrong drug. But we're doing all of these multiple tests. And when we first started doing it in clinical trials, we would send off precious tumors slides, and knowing that a panel would be done but only be told one result and have to argue to get the patient's data. It's not the company's data, it's the patient's data. But also for Ragnar, what we were doing is doing cfDNA, and if they had the appropriate mutation, we were then sending the block off. And just because for patients then, particularly ones who didn't have tumour, if they knew there was a good fighting chance they might get on the study, they were happy to have fresh biopsies. So I think we've got to work together on this, but we've got to be able to share data. And I think there's stuff coming up later on about the Primrose Consortium, but we've got to do this. So it was really a call. I mean, what do you think, Mark?
Yeah,
patient organisations, sort us out.
Completely, Ruth. I mean, I think one of the things I was thinking as we were going through this session, and sort of in my head saying, one of the things we need is to look at harmonization, but also not just harmonization in relation to the test, but in terms of sharing information, sharing data. So it might be one of the outputs of this workshop is a white paper that actually articulates that exact approach. It might be something to do with the European Cancer Organization, who also, as I sort of showed some of the data in relation to that. But certainly I think it is something that, if we could come out with sort of concrete recommendations for doing something Europe-wide together, I think that would be very useful, because I think at the moment we're not at that position, and the danger is that we all try and pursue our own line, and at the end of the day, we don't deliver for our patients. So I would absolutely agree with you, Ruth. If any of my fellow panelists want to add anything to that.
Oh, perfectly fine, but forget.
Yeah, I think, there are numerous experiences, I think, in the past by ERTC and the lung cancer work group, where there was a European-wide initiative to screen for multiple tumors and then have like a more umbrella protocol where multiple targets were available. I think those were the first initiatives. I fully agree with the previous speaker that those kind of initiatives need to be kind of looked into again, how we can move towards like sharing a broader panel, because doing the panel also from an industry perspective, it's hard to kind of go beyond the one target that we need to identify, and have such a broad tumor is from a regulatory perspective always challenging. So I think there are a lot of ways where the CCDF can we think of how to improve that. I think we're towards the end, right, Mark? I don't see the
I think that we have 8 minutes left.
Oh, we have 8 minutes left. Do we have another question online, Mariana?
I would like to ask a question myself, because I think that this is a very sensitive, the biomarker is a very sensitive topic for our patients. Still, can you suggest any strategy to make sure that this very complex concept can be accessible to patients? I know that Mark mentioned the leaflet that they produce to explain patients' biomarker concept, but I don't know if there is any other strategy that you feel like you can suggest.
Yeah, maybe I can start on this, and then my fellow panelists can also comment on it. Yeah, I think that was a starting point, but we'd be very keen on looking at other approaches that really embed patients within the drug development process. And there last year, we had a session, and we've just released the white paper recently in relation to how patients can be really embedded within cancer drug development. And I think part of that obviously is biomarker development. So I'd be very keen on exploring how we can do that, because I think one thing is producing a booklet, and I think the booklet was very useful and it achieved its specific purpose. But I think this needs to be always on. It can't be just we produce something and then we say, oh, that's great, we've produced this. And it's looking at ways in which we can work together. And certainly with some of the patient advocacy groups, at the moment, for example, it's Lung Cancer Awareness Month, and quite a number of the patient advocacy groups have been doing work that specifically relates to biomarkers and precision oncology. So I think that partnership approach, and obviously patients are an integral part of CDDF. So certainly when we're producing the white paper in relation to the activities from this particular workshop, patients will be very much involved in that. But I think looking at ways in which we can work together more and have that patient insight in relation to biomarker development, but also more importantly, its application. And one of the things we've found is I'm scientific director at JLICAN, the UK's Health Data Research Hub for Cancer. And our patient advocacy group is very strong within that in relation to looking at advising in relation to the use of data, etc. And it looks like, given what came out from Biobank UK there yesterday, that patients need to be more involved in relation to governance and decision making in relation to particularly the use of data. So maybe we can follow up with a chat after the conference to see what the best way to work together would be.
And not necessarily speaking from the pharma perspective, but from the patient perspective, I think it is a real gap. And I love the concept of having the 10 questions that people can come with. From my personal experience with my father being diagnosed with colorectal cancer, I could see that genetic testing was not a plan at his local treatment center. And, me having to ask for that to be done just showed how much of a gap there was, especially in a disease where there's clear testing to be done.
Yeah, actually, patients are the drivers of the change because most times they are very well informed. They are pushy. Also, with the doctors, that this is really useful. I think so. I would say that we find very important the collaboration with Patients Association, even to change sometimes the rules. I was telling during the lunch, we managed for the first time to introduce the word NGS in an Italian law, thanks to the collaboration with patients' organization. And there are some issues that we should work together. One of these is, for example, we know that in Europe, in most European countries, there is no correlation between the approval of the drug and the reimbursement of the test when a test is required. Now, if we should work together to get to this. Otherwise, we will continue to have drugs available and the patient cannot access to drugs simply because the biomarker test is not reimbursed. We have to think that in Italy, we adjourn the list of the tests that are reimbursed every 5 years. Now we have a new drug every 5 weeks, so it makes no sense. Now with NGS, this will be solved, but at the end, no, we will solve anyway because we test all the patients. We found a way. That's true. That's for sure. No one left behind. But we need to find a way to make this possible. And this is a simple, maybe not that difficult to get, but sometimes to change the organization is more difficult than getting the money. But this is something we have to work together because it will be the first step in order to introduce really genomic testing in the routine clinical diagnostics.
Actually, a good example, I've forgotten, I should have included in when I was speaking, is Oncotype DX in Ireland. And so Ireland per capita actually recruited most people to the trial that led to the development of Oncotype DX. But then Irish patients weren't getting access to it. So very much a coalition between Irish patients and healthcare professionals pushed that agenda forward in relation to getting Oncotype DX for cancer patients. But what was critical was that Ireland actually became the first country in Europe to through a public system to actually reimburse Oncotype DX testing, and that was through the pushing of patients pushed the agenda forward. So although they did it with healthcare professionals, the key participants and the drivers of that was the cancer patients. So that just shows you that that empowering of cancer patients can really make a big difference in relation to getting access to testing. Any last questions? We're in the last minute.
We have a last question, if you want, from Slido. Would you feel that the development of a biomarker test that qualifies as a companion diagnostic can be done by academic institutions?
Yes.
No. Very difficult because then problem is standardization. And you want to have a test that every lab in every country in Europe can run. So, I mean, I don't think that, the way is to go through academics. But, I really think in a very good collaboration between academia and companies, pharma and diagnostics early when you have the development of new biomarker. So you want a test that is reliable and that can be really be implemented in routine clinical diagnostics. And this collaboration is really fundamental.
Sorry, I think again, it's that collaboration that's important, but certainly, and particularly there are a number of now hybrid laboratories that actually are both academic and also are clinically accredited. I can think of Manuel Saltatellus, for example, in our own institution as an example of that. So I think there are ways, but the best way is collaborative approaches. Definitely, I would agree with Nicola.
I think it's a nice conclusion. We're right on time. If there's no more questions in the room, Mark?
No.
No? Okay. Well, I would like to thank the speakers very much and my co-chair, Mariana, for a great session. You really set the scene. The speakers, thank you so much for your great presentations and discussions. And I wish you all a good break. And I think we'll be back in 20 minutes online, I believe. But I think in the room, they will share that information. Thank you so much again.
Okay, so coffee break now, and we're back in 20 minutes. Is that right? Sorry, I don't have a program in front of me yet.